may join us later. Just a couple of announcements. This is the last presentation of the speaker series for this semester, which means we made it through fall, which is great. We have the spring semester pretty much all organized. We do have one date that's still a little flexible, so thank you to all of you who've suggested people. We really do appreciate it. And if your selection or suggestion hasn't shown up yet, that person or persons may come in the next year. So thank you for all your comments and suggestions. After today's seminar, there is a PhD presentation. Marine Ishmael will be defending her dissertation. She's a student of Park Wilde, so that will take place at 1:30. I think that's all the announcements. I'll go ahead and introduce our speaker. Dr. Jingtao Ma received his Doctor of Philosophy at Tufts University and completed the Intramural Research Training Award Fellowship Program at the National Heart, Lung, and Blood Institute, which is a part of the NIH. Jingtao's research focuses on the study of diet in relation to cardiometabolic diseases and risk factors, with a particular emphasis on epigenetic and protein biomarkers and diet-gene interaction. And I can just say on a personal note, he's in the office right next to mine, and he's a very, very good office neighbor. So, Jingtao, please.
Can you hear me? Okay, great. Yeah, I think because I'm very quiet, so he said I'm. I don't have much word to say before I start. Just one thing, maybe I used to sit among those seats. Couple years ago is my first time to officially present my work here. I'm a little bit exciting, a little bit nervous, maybe shaking here. So, but yeah, just let you know, and since we are kind of family, and you can stop me if you have any question. You don't have to save the question until the end of the presentation. So, and let me know if you have any questions. Okay. So I will just start my presentation. So basically, I'm going to show some of my own research, so you can know some of applications of omics in nutrition research. But this is a quite large topic actually. But I'm going to approach this topic through the angle of epidemiology or nutrition epidemiology. So you won't hear some complicated lab terminology, but more about the associations. So it will be fairly straightforward. And so okay. So for today, I will give you some examples to show you why we, at least for me, to study the omics, and also show you some of the examples so you know what the results looks like and how I interpreted those observations. And also what the challenge for each type of the analysis, and maybe I can add, and with what the future directions for those type of analysis. So I assume most people know the omics, but just for people who don't know, I quote these things from the Wikipedia. So omics informally refers to the field of study in biology ending in omics. So it's quite straightforward, just omics. And it's, for example, like genomics, proteinomics, metabolomics, and what types of omics. And the omics aims at collectively characterization and quantification of pools of biological molecules that translate into the structure, function, and dynamic of an organism or organisms. So that's a large picture for omics. For me, my goal certainly is apply the nutrition epidemiology to understand how diet interact with our genome and interact with all those omics markers to understand the mechanisms underlying the diet association with diseases. My focus is more on cardiometabolic diseases, but the approach can also be applied for other type of diseases. For example, like the Alzheimer's disease, those neuron degenerative diseases, the people using a similar approach. Okay. So since I said I focus on the cardiometabolic diseases, I want to show you why those diseases are important. Here you can see, I have a pointer here. So basically here you can see the heart disease, stroke, and diabetes. Those are major cardiometabolic diseases. They are among the top killers in the American people. So especially the heart disease, although the number of absolute heart disease deaths is decreased, but still is the number one killer in the United States, and actually is also in many other developed countries. So that's the reason we want to know the mechanism about how this disease developed, and so hopefully we can find some treatment to cure disease or to prevent this type of disease. Okay. So actually many study has been done to focus on different kind of omics to study the biology of heart disease. So this plot basically shows the central dogma of biology from the information, just demonstrate from the information from DNA to the phenotypes. The phenotypes basically diseases or a trait like BMI, so we call it phenotypes. So you see the information from our genome can translate transcript to gene and then translate to protein, and the protein can be metabolized to all kinds of metabolites, and then have functions on a certain phenotype or cluster of phenotypes. So this type of analysis is quite useful to understand the biology for diseases, especially the genetics, because the genetics, basically for all those omics markers, now the technology allow us to measure them in a kind of robustly measure those markers, and also it can give you high throughput outcomes. So basically you can measure them in a large quantity of those markers, and you can analyze them quite robustly. And so I just emphasize the genetic is more important because the genetic is not confounded by environmental factors. So even though the effect size on some complex diseases is small, but that association is kind of not confounded by other factors. So for sure those factors are causal factors for certain diseases. So I will show you. Here is a plot I just downloaded from the GWAS catalog. It shows the genome-wide associations. There's a significant findings from genome-wide association studies. This is a type of common. This is a common approach used in the omics analysis. So basically, just analyze all those markers in our genome or all those proteins coded by this genome in association with phenotypes. So this plot basically I downloaded a couple days ago, so is update until November. So it's included about 8,000 papers and 20,000 significant associations. So each those ideal graph or small graph just shows one chromosome, and each colored dots here represent a genetic locus, and there is a significant mutation has been linked with a phenotype. So up to perhaps today, there are over 1,000 SNPs has been associated with CVD. So the SNPs, just a abbreviation for a type of genetic mutations. It's called the single nucleotide polymorphism. So basically, have a picture here. Basically, say for one person, if they carry A allele at these genetic locus, and the other person carry a G allele, so basically arginine and guanine in different in same locus. So we call this is a single nucleotide polymorphism, or SNP. So basically a genetic mutation on a single base pair resolution. So GWAS is useful. And here show you some examples why GWAS is useful. For example, the GWAS study identify a genetic mutation at this gene is a solute carrier family 30 member 8. So this gene actually is called a protein called a zinc transporter protein. It is located in the pancreas. So the variation of this gene can affect the pancreas performance. So this another type of analysis, they work together. So leads to a development or discovery of a drug can have a function to lower the blood glucose. So this is kind of a the shows the usefulness of the GWAS. And the other example I want to show you is the GWAS identify genetic mutations next to this gene. This is a well-known gene can affect the lipid level, especially the low density lipoprotein. This is the HMG coenzyme A reductase. So this gene is well known long before the GWAS era. So a lot of statin actually work on this enzyme. A lot of statin medication. So they lower LDL because they are kind of how to say that is work on these pathways and to block these enzymes so they can lower the LDL. So for GWAS, when they identify a mutation for these genes, so you can use this mutation, these SNPs or this genetic mutation to look their association with different phenotypes. So you can do a phenotype-wise association study called FAVAS. It's become very popular. So you can run a FAVAS and to figure out if these genetic mutation can be linked to another phenotypes. So basically, it can tell you two things. One thing is whether this mutation can, or this you can develop a molecule to used for other indications. So basically, it's saying if you can use statin for other type of indications or some off-label use other than lower LDLs. And the other use is you can use this information to test whether statins have any side effects. So because most of these trials focus on lower LDL, but when you run the FAVAS, you can know whether certain medicine can have some of the side effects we don't know when we do the clinical trial for these drugs. So that's a very popular thing now. they do this type of FAVAS study to look the, after the GWAS, you identify some, genetic mutations. So those are two major things now for GWAS study. And however, like I just mentioned, for a lot of complex trait, the GWAS can only explain a small amount of inter-individual variations. Here is one example for a very common trait, the BMI, the body mass index. So this shows the latest GWAS study was published in 2018, it's, last year, and they basically meta-analyze multiple GWAS results. This study was done in about 700,000 participants, a very large GWAS study. They, done a GWAS in UK Biobank, and also they get the summary result from a previous GWAS study published, I think is in Nature. And so they basically meta-analyze those results together, and this study pushed the SNPs identified or associated with BMI to about 941 SNPs. Those are independent SNPs. So the study in this GWAS paper, their number probably is around 100 SNPs. So they substantially increase the number of SNPs for BMI. However, you can see here for the variation of BMI, this blue bar shows the latest GWAS. It only can explain 6% of variation for the GWAS for BMI. If you use a more relatively liberal threshold, use 10 to the minus 3 as a very liberal p-value threshold in GWAS. So you can find about 6,000 or 7,000 SNPs. They are independent SNP. I mean, like by independent, so they are the linkage disequilibrium or LD is below 0.02 or something. They use that threshold. So even you have 7,000 SNPs, you only can explain nearly like 14% of a variation for BMI. So that means a large amount of a variation for BMI for this common trait cannot be explained by these SNPs identified by those GWAS. Even you have very large sample size. This actually also true for other common or complex diseases. For example, for systolic blood pressure, I didn't show here. Now the systolic blood pressure GWAS sample size is about 1 million. So they combine the UK Biobank and the International Blood Pressure Consortium, and also part of data from the Million Veteran Program. So about 1 million people, and the common SNPs identified by those GWAS can also explain about 5.9 or about 6% variation for systolic blood pressure. So that means maybe diet play a role here. But the other thing is I want to tell you is there are also many other type of genetic variants. For example, here they focus on the common variants, and there are other type like the rare variants. So the low frequency mutations also may explain a certain amount of variation, and other type of variation like insertion or deletion. Those different type of variations, they may be able to explain another part of variation. And also gene-gene interaction now is very hot topic, so people start to working on that. But most people predict the environmental factors and the gene environmental interactions will be explain the larger part of a variation for these common traits. So relatively to the common traits, GWAS signals can explain a little bit more for biomarkers at molecular level. Here shows one example for proteins. So the y-axis basically a list of CVD-related proteins. You don't have to know the names, but just CVD-related proteins. And the x-axis basically shows the inter-individual variations from 0 to 100. So you can see here the pink colored bars are SNPs, and they for many of proteins they can explain more than 20% of variations. So it's certainly larger than the complex trait like BMI. And for many proteins, they can explain more than 50%, so the middle line. And for some proteins, it can explain like more than 80% of variation for protein markers. And this study basically is done using the data from the Framingham Heart Study. And they also look some of the clinical variables and demographic variables like sex, age, smoking, and type 2 diabetes, and those kind of markers. And these can explain a little bit variation for these proteins, but the gray bars basically are unknown factors, and the diet is among those unknown factors because this study doesn't include the diet as one factors. So this is gave me and some other people a hope, like maybe we look the diet and in association with this omics or interaction with this omics, maybe we can find some interesting findings from that. So that's basically I'm doing now is look on the diet and look how the diet interact with genome and with other omics. So because diet is this not only because they didn't find enough variation by the genetic markers, means diet can be a factor. Actually, there are many evidence have shown that diet, it can be a factors directly or indirectly, work on those either genome or other, omics and have certain functions. So that's a kind of a premise for why I'm doing this type of analysis. So by doing the nutrition and omics analysis, my hope is, basically I want to know the exact association between diet and diseases, whether this association depend on certain, for example, genotype or other omics levels. And also, I want to know, the mechanism how diet can, affect or have any function on certain phenotypes. And the other thing is, by looking by add a diet as a factor into the model, you can maybe able to find some of markers, either genomic markers or other omic markers, maybe able to predict the diseases, or maybe to provide some insights for future analysis. Maybe we can able to find some therapies for certain diseases. So those are three goals I'm trying to achieve. And here is one a simple example how I did a diet gene interaction analysis. So in this analysis, it was published in next year. So basically, I'm looking for the change of the diet quality, and whether that change of diet quality can affect the change of liver fat or the risk of develop non-alcohol fatty liver disease. So the x-axis is basically a change of diet quality. I use Mediterranean style dietary score as a proxy to reflect diet quality. I also use other scores, but here just show you one example. So there are three panels. The left panel shows the people decreased the diet quality over time. And the middle panel, basically their diet quality maintained, and the right panel just shows their diet quality increase over time. And the y-axis is the change in liver fat. Here is a little bit confused here, like because we use CT to measure liver fat. The CT image is kind of inversely associated with liver fat. So you have a higher kind of image. Here we use ratio, the liver to phantom ratio. So if you have a higher ratio, means you have lower liver fat. So here means the liver fat increase, and if the lower is going down, well, the negative number means the liver fat increase. So, and the different color represent a different genetic background. So I calculate a genetic risk score for those participants. So the red color means they have high genetic risk, and the orange means the middle risk, and the green color means they have low risk. And those genetic risk score based on the GWAS findings. So basically I use the summary result and also measure the genotype in the Framingham heart study participants. So then I can calculate the score. And here, if you kind of summarize, you calculate the mean of these 3 points. So you can see for people increase their diet quality, their liver fat, basically, they only increase a little bit. And this is also same for the people maintain their diet quality. You see a little trend, but doesn't significant. But here in the people decrease their diet quality, you can see, a clearly trend, according to their genetic risk. So for people have a higher genetic risk, and their diet quality also decrease, their liver fat increase the most. And for people have a lower genetic risk, and even though they have their diet quality decrease, their liver fat doesn't change very much. So that's the finding from this analysis. So we formally test the interaction. We also found a significant diet gene interaction here. So this is one example. So in another study, we basically repeat this analysis, but we replace the pericardial fat, replace the liver fat by the pericardial fat. The pericardial fat basically a adipose tissue, around the heart, so we call pericardial fat. And here in this y-axis, basically the higher means a higher liver fat, and lower means lower liver fat. So I reverse the order for this pericardial fat. And the x-axis is basically the same. It depends on the diet quality change, and also depends on the genetic risk for the pericardial fat. Here basically you see the similar pattern here. So for people decrease their diet quality, and they have higher genetic risk score for pericardial fat, they have higher increase of their pericardial fat. For the other 3 categories, you didn't see much of an increase in terms of pericardial fat. And actually for people have lower risk, genetic risk, and also have higher increase in their diet quality, they have lowest they decreased most for their pericardial fat. But this study, we didn't see any significant interaction when we formally run the interaction test. So one reason we didn't see that, it perhaps because the genetic risk score. And for the liver fat, the GWAS study did a relatively good job. But for this GWAS study, they didn't provide the effect size for each SNPs. And also perhaps that's probably because different cohorts, when they meta-analyze, like I think it's 4 or 7 cohorts, and different cohorts use different approach to measure visceral the pericardial adipose tissue. So you cannot really meta-analyze those data together, give you a quantified effect size. So they don't have the effect size for that. And the other thing is, For this GWAS study, for actually both liver fat and adipose tissue, the sample size is much smaller than the GWAS study for BMI. For BMI, you can see the number is about like 700,000. For the liver fat GWAS and also for the pericardial adipose tissue GWAS is only 7,000 people. So the number is just reduced because the CT is very expensive. So you cannot really do a robust GWAS for all trees you are interested. So that's a kind of limitation restrict us. We cannot build a robust genetic risk score to do this type of analysis. But that's kind of one challenge. But the other thing is you don't have to really to calculate genetic risk score. You can run a genome-wide or candidate gene diet gene interaction. So, but still you will be limited by the sample size, and especially when you run a genome-wide diet gene interaction. So now for common measure genetic variants, you can measure about 2 million SNPs, and those common SNPs, and some cohort you can measure a little bit more. For the UK Biobank, since they have larger sample size, when they define common SNPs, they define the minor allele frequency by 0.001 or 40. So the number of SNPs are even higher. So I see some report they can they have about like 16 million SNPs they consider common. And so consider those the multiple testing you have to run. So you have to pay the penalty for so many tests you conducted. So that's also a factors limited the genome-wide diet gene interaction analysis. And also for diet gene interaction per se, you have to you need a larger number of samples to get a significant result. So normally, people say you need 4 times of a sample size to get a significant result compared to just normal association analysis. So that's a kind of a major limitation for diet gene interaction analysis. But still, it's a very interesting concept, and we believe diet gene interaction is a major contributor or factors to decrease or increase the risk for diseases. So we so here people developed a model to allow you to look the diet gene interaction or diet or gene environmental interaction, and meantime you have a larger power to identify some of new genetic variants or new genetic loci. And someone called this Manning approach, but actually it was first proposed by the Peter Graf over Harvard. And basically, you in one study you look both the main effect of genetics and also look the interaction effects of a genetic with environmental factors or for example for diet. And then using the model, the formula they provided, you can calculate the joint effect for effect size and the standard error. So this way, so even though you cannot see any significant effect for the interaction, you may be able to find some significant effect for the joint analysis. So I will show you one example here. In this analysis, this basically a preliminary result I get using the data from Framingham Heart Study. The sample size is about like 5,000 people. It's considered very small. And right now I'm working on a larger project with, in collaboration with many kind of a community-based cohort studies to see if we can replicate findings in Framingham heart study, and also we can, basically increase the sample size. So here, I didn't see any significant significant result for the interaction analysis, but in terms of the joint analysis, you can see some SNPs at chromosome 11 and also chromosome 5 and 4. This is some interesting result here. Basically, they are kind of significant in this smaller set of data. One more thing is this is called Manhattan plot. So someone maybe know that, and basically the x-axis are chromosomes. So from chromosome 1 to chromosome 22. So it's all autosomal chromosomes, and the y-axis is just minus log 10 p-values in terms of the association studies. So people just call this Manhattan plot. It's very common in genetic or other association studies. So the other plot I want to show you is the comparison between the main effect as x-axis and the joint effect, on the y-axis. So here you can see a lot of each point represent one SNPs. So along the diagonal line here, you see many SNPs are aggregated here, and for those SNPs, actually the main effect model have a slightly lower p-values below the diagonal lines. So basically for these SNPs, it's saying there's no genetic or diet interaction. Here the diet, I use a dietary score here. So, and their p-value a little bit higher actually, the higher p-value means not significant, right? So a little bit higher in the joint analysis, because you have in the model you have one interaction term that, so you have to pay the penalty for one more degree of freedom. So that makes p-value a little bit higher. But the interesting part is here in this area, you can see many SNPs, they are not significant in the main effect model, but they become kind of significant in the joint effect models here. For example, one SNP, this SNP is a second significant SNP in this analysis, and this SNP is located in the chromosome 5, and is the reference number for this SNP is this. So it's close to a gene called CDH18. This gene is a calcium binding gene. In the previous GWAS studies, they found a SNP next to these genes associated with systolic blood pressure, and but that SNP is far away from these SNPs. Is about like the distance between two SNPs is about like 1 million, is more than 1 million base pairs. So it seems, in this, even in this small set of data, we found a kind of new SNPs here may have a function, may interact with our diet and have a function on systolic blood pressure. So I hope this is the result is true. But I'm trying to replicate, as I said, in many other cohorts. But this is one example. The joint analysis can, help us to identify some new loci. Actually, there is another group people. They are working on this, using this model to look the diet gene interaction, and they have found some very interesting findings in terms of lipid levels, and they just published in the Nature Genetics early this year. So hopefully we can use models and introduce the diet in the models and to find more findings here. Okay, I need to hurry up. I'm just trying to switch gear to another topic, and this is plot shows the epigenetic regulations, and the epigenetic regulations basically is different from the genetic mutations. So genetic mutations basically tell you the change of the sequence of the genome. The epigenetic regulations doesn't change the sequence of the genome. It's only add some compound or affect the structures of the genomes. For example, there are three types of epigenetic regulations. So one is the RNA-based mechanism. So for example, there is a microRNA, this small piece of RNAs is also coded by our own genomes. Those small RNA, the microRNA, can be released, and it can regulate the gene expression levels. And there's also other type of RNAs, like long non-coding RNAs. It now is also become very hot in this area to look how that long non-coding RNAs to regulate gene expressions. And the other major type is histone modification. So basically, just the histone modifications, the structure change. It can, change the chromatin. So basically, it will affect the gene expressions. And the things I studied most is DNA methylation. So it's also a simple, straightforward modification. It just add a methyl group on the genome. Here is one picture. So basically, is on the 5 carbon of cytosine, there is a methyl group attached to here, and so this cytosine become a 5-methyl cytosine, and these can either increase the gene expression or decrease the gene expression. And the earlier study shows the methylation tend to decrease gene expression, but now more and more studies shows the methylation can increase the gene expression. So we call this DNA methylation site a CPG. So basically, a cytosine follow a guanine, so always like that, and so we call it CPG. Here I conducted a kind of a epigenome-wide study. It's a similar concept for GWAS. It's also the whole genome-wide association analysis, but looking for the association between diet and the CPGs basically. So this was done in 5 cohorts in the CHARGE consortium. So this is large consortium, basically allow you to conduct a meta-analysis and to look for either GWAS or EWAS or other omics analysis. So after we identify some significant CPGs, I also done some bioinformatic analysis I will show you later on, and also look for the longitudinal analysis between the CPGs with all-cause mortality. Here is one result for the EWAS of a Mediterranean-style dietary interaction analysis. It's not like a GWAS. We didn't see a lot of high spike here. It's because for GWAS, you have like 2 million SNPs or 8 million SNPs, but for CPGs, here is only 450K SNPs. And right now, you can use other array. Maybe you can measure like 900 CPGs, and you also can use the whole genome sequencing approach. You basically can measure more SNPs, but the 450K array is kind of a low-cost approach. But regardless the approach, you see there are some signals here associated with the Mediterranean score. And for some score, for example, for CPG here is annotated to a gene called SOCS3. And these CPGs, when we look the longitudinal data, is associated with all-cause mortality. And those data we use, we put actually the result from 10 cohorts, about like 10,000 participants. So quite robust association here. It says it can higher methylation at this CPG significantly reduce the all-cause mortality in these 10 cohorts. And when we look deeper for this gene, we found this SOSC3 gene, kind of inflammation genes. It can repress the release of many cytokines. For example, the IL-6 is a common inflammation markers. Have been shown with many other diseases, including the heart diseases. So that may tell us diet maybe regulate the DNA methylation and affect the inflammation and affect the downstream diseases. So another example is also another CpG is at chromosome 9 here, and this CpG is annotated to a gene, basically annotated 2 genes, and the AQP3 gene can regulate the water channel, and it has been linked with blood pressure and also with some heart diseases, for example, congestive heart failures. And this also gave us some hint for how diet maybe affect the diseases. But these are all observational data. So we trying to, we cannot do a function study here. So we're trying to, use observational data to give us more information in terms of whether these CpGs, what these dietary related CpGs are causal factors. So we do this longitudinal analysis, just give us more information on the temporal relationship. And we also run a Mendelian randomization analysis. So basically, this type of analysis mimic a clinical trial or randomized clinical trial using the genetic variants. So basically, using SNPs here, we want to know if the CpGs are truly causal risk factors for a certain disease. Here, we're looking for CVD risk factors, for example, BMI, type 2 diabetes, lipid levels, and so on. So one assumption here is the SNPs cannot be associated with a confounders, and also cannot directly has a function directly effect on the CVD risk factors. So it has to go through the CpGs. So it's hard to prove those is true, using the observational data, but we try to use the SNPs called cis MEQTLs to as a instrument variables, and the picture here shows what is cis MEQTLs. Basically, are SNPs close to a CpGs. By close, I mean they are distant. They basically are the same chromosome. And so the distance wise, so they are less than 500 kilobase per away from the CpG. So that means close, and also you run the association analysis, and to see if that SNPs associated with these SNPs, these CpGs. So basically, all those cis MEQTL, they have an association about 5 times 10 to the minus 8. So they have very strong association with these CpGs. So we use these as instrument variables. So basically, we group our participants depends on their SNPs, depends on their genomes. Someone have higher CpG levels, someone have a lower CpG levels. So it's something like a randomized clinical trial. So this is basically based on the Mendel's second law, the law for segregation. So there are a lot of theory behind of this Mendelian randomization. It's useful to show the causal association, but still is a observational study. It is not for sure, but it gave us a little bit confidence to show whether some CpGs are causal. So here are just 2 examples, and the left picture shows the CpG here, maybe a causal factors for triglycerides, And the other CPG may be a causal factor for BMI here. So basically, you have information on these SNPs, their effects on this CPG, and you have the SNP effects on the triglycerides, and then you can calculate. The analysis is pretty straightforward, but it gave you a lot more confidence to show the markers we identified in this EVAS study, kind of causal factors for CVD risk factors here. Okay, I have a couple of slides for protein analysis. Maybe I will give you more time to ask questions. I will just keep just one slide. So we found in a study. I'm just still working on that analysis. Basically, looking for again dietary scores in association with a bunch of protein markers. Those protein markers are all CVD-related proteins based on either GWAS study or expression study or the protein omics studies. So we found a protein called GDF-15 is significantly associated with the dietary scores, either Mediterranean score or DICE score or AHEI, those different kind of scores. And this protein can mediate the association between the dietary scores with all-cause mortality. So the mediation is about like 30%. So basically, it can explain a very large amount of association between the dietary scores and all-cause mortality. And we found, we saw this protein may be an interesting protein, and we looked at the protein-protein interaction networks. And this we got this network from the String website. They had a database for protein-protein networks, and some study shows this GDF-15 can work on the receptors in the brainstem. This is a recent analysis, and they show that this activation of the GDF-15 receptors can promote pathways called non-homeostatic pathways, and can increase the food intake and also have effect on body weight. But those pathways are not typical pathways we use to regulate dietary intake. It's normally upregulated in the situation like high inflammation occurs or in some disease status. So it's not typical pathways. So maybe that's an interesting findings. We don't know, and we also look for we build a genetic risk score based on the GWAS study for the GDF-15, and we look the association with all-cause mortality and CVD. And like I said, this protein can mediate the association between diet and all-cause mortality. But here we didn't see anything, this genetic risk score associated with either all-cause mortality or CVD. And that's kind of things we need to deal with, and whether or not this protein a truly causal protein for heart disease or other disease, and the genetic analysis doesn't provide robust evidence. But like maybe you remember, I showed at the very beginning the proteins can be determined by genetic factors and also by other factors. So maybe here we didn't see any significant association. It's because the genetic variance doesn't really explain a large amount of variation for these proteins. So certainly, data here from the observational cohorts cannot answer that question. We need more data from functional analysis, and hopefully, we have trying to collaborate and trying to figure out if we can run either cell studies or animal study to show whether the proteins were a causal protein or not. So the takeaway. So hopefully you probably I'm speaking too fast. So hopefully you get this message here. So by looking for the diet and omics relations or interactions, you can have a better estimation for diet disease associations. You can, find some mechanisms in terms of diet, how diet affect diseases, and you can, identify some novel genetic markers or omics markers and give you a hint for, disease predictions, or, hopefully you can find markers to prevent or treat diseases. But for the future directions, in terms of, I just mentioned, like for some GWAS, we may be to do more GWAS studies for like phenotypes, for example, like visceral adipose tissue or liver fat or other markers. It's not as common as BMI, so we need to do more that. And also, one thing we need to do is certainly to have a better quantification for diet. Regardless of the genome-wide is genomics or genetic variants or other type of omics, you have a pretty robust approach to measure those markers. But in terms of diet, we only have a FFQ or maybe other 24-hour recall, and those suffer the measurement errors. So when you run the association and interaction analysis, the power will be reduced because of the measurement error. So we need well-quantified dietary data, and also we need some novel computation approach. For example, for genetic risk score, now people have a different approach to not only use the significant SNPs identified from GWAS, they can allow you use all the SNPs from the GWAS studies. So you can use perhaps 10,000 SNPs to create a polygenetic score. It can have a better prediction for diseases, and then you can use that score to look the diet gene interaction, maybe give you more significant findings. And also, in terms of all these analyses I show you here, we not only do a data analysis in single cohorts. We basically do the similar analysis in multiple cohorts, and then we meta-analyze all those results. So, but different cohort, they use different approach to measure omics, measure diet. So we need a good consensus or good approach to harmonize those data, to standardize those data, so we can get robust observations from there. And also, like I just mentioned, in terms of for protein example, that we need functional study to validate our analysis just from the observational studies. So, okay, the last slide is acknowledge my mentors and collaborators from those cohorts, and thank you so much for your.
We have time for maybe one or two questions, if there are any.
Thanks. So that was really, really interesting. You went over so much. I have two questions. The first is, when you were doing this study where you're looking at fat percentage in liver, it looked like there was a increase in liver fat when you saw a decrease in Mediterranean diet score, and I was wondering over what time period that diet change score happened, and whether or not there was any weight loss associated with it. So what was it the diet change that was driving this downward trend, or changes in weight potentially?
Yeah, if you calculate, you basically if you look the mean of these the change of liver fat, actually you see like the diet can have an effect. So here the liver fat should be like at here, and this is a little bit higher, and this is even higher. So diet certainly have an effect on the liver fat change over time, and the gene also have an effect here.
But I'm wondering if there was anything else going on, like as the diet changed, was there any weight loss?
We in the model we adjust for the baseline BMI, and also we adjust for the change of BMI.
Okay.
So
independent of any weight loss.
It should be, but if something is really correlated, like the liver fat it should be correlated with body weight. So if something is really correlated, it's hard to adjust in the model, but we did adjust the model. So I'm pretty comfortable to say that, but we certainly need more robust data to show that. Yeah.
Okay. And one more question I had, I think it was a slide a couple before this where you showed there was like lots of different color bars.
Okay.
That showed like the contribution of different SNPs as well. Yes, this one.
Oh, okay, this one.
No, yeah, this one. So I wonder, so with all these gray bars where you say other factors could be associated with CVD risk, I wonder, you were talking about how diet can be part of explaining those other relationships. I wonder about physical activity, potentially stress hormones. Is there a benefit to looking at diet, physical activity, stress, all of those things together in order to get more specific estimates of the effects of all those? Or is it okay to, like, I'm wondering if doing a study altogether is going to give you more specific and valid estimates of the contributions than if you were to look at the contribution of diet that fills in those gray, versus, as well as in a separate study looking at physical activity. I mean, it's difficult to look at everything together, but are there advantages to doing that?
I think it certainly has an advantage to do that. Actually, tomorrow at Framingham there is a talk just looking the interaction of diet and physical activity in terms of heart disease. I mean, it's very interesting to look everything together because my major thing is diet, so I try to present the diet. But certainly you can do those together. But one thing is, diet is already very hard to measure. Physical activity is even harder. That's my understanding. So I mean, certainly it would be more than interesting to look multiple factors. So if we can do it, certainly want to do that. Yeah.
Thank you.
Thank you. We are at time, and there is a class that comes in afterwards. So let's thank Dr. Ma. This completes our seminar series for the semester, and we will see you back in January. Thank you all.